as further evidence of Oxford being a good place to be if you have Parkinson, we have Professor Richard Wade-Martin. Richard leads the team at DPAG, as we always call it, Department of Physiology, Anatomy and Genetics, a major part of the Oxford Parkinson's Disease Centre. And as Ellie and Yatin, who are here, two of the regular visitors from DPAG, they're tremendous supporters. And meanwhile, great work is being done down in the science area, funded in no small part by Parkinson's UK, some of which money comes from what we raise. So, Richard, thank you so much for coming. We're very pleased to have you, and I'll hand over to you.
Thank you. Thank you very much for the invitation to come here and tell you a bit about our work. Thank you, as ever, for the kind words and warm welcome. It's very nice to be able to come here and tell you a bit about the sort of things that we do. We try and bring a couple of people along each month to come and meet you. And I'm very pleased that Ellie and Jatin have come along this evening, and they can answer all the questions that I can't. Well, what I thought I'd do is just start by giving you a brief introduction to some of the work that we've got underway, an overview of the Parkinson's research that we have underway at Oxford, and then move on to the topic that the title of the talk, Why Do Brain Cells Die and What Can We Do About It? So first of all, a bit of an introduction to the Parkinson's research and discovery science, the sort of fundamental lab-based science that goes on at Oxford, a lot of which goes on in our department in physiology and anatomy and genetics. So we've put together, I'm tremendously proud of what we've been able to put together with a lot of the support from Parkinson's UK, a genuinely multidisciplinary translational neuroscience program that we started in 2010, myself in close collaboration with Michelle Hugh, a fantastic movement disorders consultant. Many of you will know. We've put together some of the best exemplar, I think, of what Oxford can do when it brings together the range of experience and expertise that exists in scientific laboratories and in the hospital setting. And so this Oxford Parkinson's Disease Centre, we started in 2010, going about 14 years. The main achievement is we all still like each other, which is quite something. There's about 10 of us involved, scientists and clinicians, and this slide will evolve and open to show you the three kind of key aims and themes of the work. So first of all this sort of work starts with working in close collaboration with patients, friends, families, carers. And Michelle has put together this cohort of about 1,000 Parkinson's patients from around the Oxfordshire region. It's a Thames Valley cohort, sort of Northampton in the top right and Reading in the bottom left. It's a bit of a sort of stretched and extended Thames Valley, but it's everybody's about a 1 hour or so drive from Oxford, and there's regional hospital centres where many of the cohort, many of you have been involved, have been followed. So Michelle and her clinical team have studied about 1,000 individuals with Parkinson's over 10 years, and every 18 months we'll be visiting Michelle and her colleagues and have a whole range of tests and being poked and prod and measured for all sorts of things. But it means it's now a fantastic, what we call a longitudinal cohort. So individuals tested for a whole range of clinical movement and motor and non-motor phenotypes features over the last 10 to 12 years. And it's just a brilliantly coordinated a longitudinal set of data that Michelle has coordinated. And it's the point of the cohort is what we call for biomarker studies. So Michelle has in her freezer blood, plasma, serum, CSF, all sorts of samples collected from the patients, some of which go back for 10 years. And Michelle says, Richard, when you've seen one patient with Parkinson's, you've seen one patient with Parkinson's. Individuals develop in a very different way. And the idea is that if we follow clinical progression of individuals in the clinic, and then we have the samples of blood and plasma, serum, CSF in the freezer as well, which we can follow as well, it would mean that in future work, we'll be able to go back to some of the very first samples that were given, because we then know how that individual developed 14 years later. So would you be able to predict from the very first samples how the condition will develop? So Michelle's built up this fantastic resource. And my own lab is a cell and molecular biology lab. So we work with patient cell systems that we've taken. So individuals have donated a skin biopsy, which is then taken back to the lab. The skin biopsy is processed, and these cells called fibroblasts will grow out from the skin biopsy. And these can be reprogrammed with some molecular tricks that I'll tell you about a little while later turn the fibroblasts into stem cells. And when you have a stem cell from an individual, you can make that stem cell into any cell type you want. So we're interested in making dopamine neurons, these brain cells that are dying off in patients with Parkinson's, to understand how dopamine neurons from healthy individuals, as from people with Parkinson's, how we can study them in the lab. So I'll tell you about some of these things that we're doing. We work with what's called phenotypic screens. So a phenotype of a cell is its features and some of its biochemical properties I'll tell you about. And we can work now with groups of cells and single cells, and we can put together cells in different multicellular systems. So we can put different neurons together. And not then, of course, they're not mini brains, but we can start to build circuits of neurons together. So theme 2, advanced cellular models made possible by the skin biopsy donations. And the third component of the program is where we work with mouse models of Parkinson's. So Parkinson's is a disease that affects humans, so we work with patients and we work with cells, but also we need to know how a whole brain, the function changes if you take out these dopaminergic neurons. If you lose the dopaminergic neurons, how does that affect the brain? And the mouse models have been very informative on telling us how the brain functions and changes once the dopaminergic neurons are lost. And we've got some of the best world experts on how this part of the brain, called the basal ganglia, how the connections that control movement, how they work. Some of the best basal ganglia experts in the world have helped to build the program with us. So the idea is you then wish to complete the circle. So we study people with Parkinson's, we study the cell culture models, we can test for new drugs in the cells, and ultimately we hope to find new potentially therapeutic compounds, test them on the mice that have Parkinson's, and then ultimately we'd be able to feed that back into a patient cohort. And it's been a tremendously busy but brilliant opportunity to collaborate. We've got about 10 departments right across Oxford, uphill, downhill, the uphill clinical departments, the downhill science department. We work with colleagues in Bristol, colleagues in Cardiff, many places around the country to put together, because this, whether you're working in an academic setting or for a pharma company, this is what you need to tackle a neurological disease. You need a well-defined patient cohort, some great cell biology, and some in vivo, in-brain physiology as well. So we've been going 14 years. What have we achieved? Well, we've established the Oxford Parkinson's Disease Centre. It really is an international centre of excellence for Parkinson's research happening right here in your hometown, recognised now internationally for our work. Particularly, it takes a long time to get the Americans to realise that great research is done outside America. But our American colleagues now who know who we are, we get funding from the Michael J. Fox Foundation in the States. It's just taken a while to get recognised. The discovery cohort data and biosamples that Michelle has collected, and many of you may have contributed to, the stem cell lines that we've made, and the transgenic mice and rats we made are now in use across the world, in academic and also in pharma labs as well. So some of the mice, for instance, we've now made are in use by pharma companies who think they're some of the best mouse models of Parkinson's. We have brought together clinicians and laboratory scientists together in an integrated program of translational work. You cannot believe how hard it can be to get people who don't quite understand what the other does together in the room together, and you build up trust, you build up friendship and a collegiate way of working. We've done it really well. We said we would, and we have trained the next generation of Parkinson's researchers. So I was speaking at a meeting in York about 3 weeks ago, a Parkinson's UK and funded program for scientists across the country. And there were 5 people in the audience and speaking who'd either come through postdoctoral researchers in my lab or at the centre, who now have their own fellowships in Cardiff, and he's a consultant pathologist in London, and just, many people doing great work nationally because we trained them here, and that's really good. And throughout this whole program, we have benefited fantastic, very strong support of the local Parkinson's community, and that's you. So thank you very much for your support along the way. Things that have developed and new things when it comes on the screen. My own lab moved in 2021 to a new research building called the Kavli Institute for Nanoscience Discovery. So nanoscience is anything that's very small. So it's a slightly ambiguously worded center, but the vision is that it's a new research laboratory, new research center on South Parks Road, just behind the Natural History Museum. And the point of the center is that it's a lot of cell biologists like myself and my team in this building, along with people who do physical sciences things, so medicinal chemistry, bioengineering, electron microscopy. They do lasers and stuff and things we don't quite understand. But we're building up collaborations with people who do these techniques, because the point of the center is to bring these physical sciences techniques to focus on the cell of choice, and the cell that's been chosen is the neuron. So that means that there's a lot of people around us who do these biophysical techniques we don't quite understand, but we're getting to know them and getting to work together. And it also means there's many people in the center who also work in stem cell models of brain disorders. We've got colleagues who work on Alzheimer's and motor neuron disease under the same roof using similar techniques. So it's a great collaborative environment. So this is what the building looked like in 2019. So we had we were working in the building next to it, and for many months, we peered down into this big hole, which was kept from falling down by these large yellow jacks. And because it was a new building that we moved into, and we knew we were going to move into it onto the ground floor, I helped design it from scratch. So the labs, the layout, the equipment rooms were all laid out for us. It's fantastic. And then that's us in February 2020, 11th of February 2020. I don't know if you can remember what happened in February 2020. This was almost the last thing we did before total close down. And this is us in our office area, delighted and thrilled to be having a look around. And many of these people are now, he's now an independent fellow at Cardiff. She's an independent fellow at Cardiff. Charmaine and Brent, many of you may have met, have fellowships now in my lab in Oxford as well. So it's really nice. So things sort of stopped for a while. Well, the building kept going, but we didn't visit it for a while. And then we followed it, and this is October 2020, it was being put together. Then we finally got to move in. So in June 2021, we moved out of where we were. It's just like moving house. All these boxes arrive, and there's chaos for a week or two as you pack everything into the boxes. And then the other end, you unpack it. And, you know, Ellie and Jacqueline will tell you the stem cell lab does not look that tidy anymore. This is the tidiest the labs will ever have looked. And we unpacked and moved in. Big celebration. And it took a while for sort of us to fully settle in. This is how my office appeared for a while with the building site just outside it. But it's a fantastic opportunity. And as an academic, you always like to grumble about not enough space and not enough money and da 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 da. We now have lab spaces that are absolutely brilliant. So that's great. So that's a bit of an introduction as to where we've got to so far. So the next thing is to think up what are the future strategic directions for Parkinson's research at Oxford. I have many inspirations in my life, and this is one of them. Who's that? Or in character? This is Thomas Shelby, right? Absolute hero of mine. He is a psychopathic Birmingham gangster, but apart from that, he does have some great ideas, and he knew how to work out what to do as the best in the future. This is one of my favorite quotes from him. He said, "My father told fortunes and he stole horses. Often he would tell a man that his horse would be stolen, and they would marvel at his powers when it was." So he was very good at predicting the future and, you know, the best thing to do. So obviously it's harder to work out what are the future strategic directions for our work at Parkinson's in Oxford than it is for Thomas to predict the future. But here's my thoughts as to some of the things we're doing and where are they going. So a lot of our work is understanding why these brain cells, these dopaminergic neurons, die in Parkinson's, and how we can advance technologies and methods to work out how to save them. So as a brief introduction to what's happening in the brain with people with Parkinson's, we have a loss of a particular type of neuron called the dopamine neuron that makes dopamine. The neuron starts in this region called the substantia nigra and sends its projections into the striatum. And they make dopamine and release dopamine into the striatum. If you lose the neurons in the substantia nigra, you lose the ability of dopamine to be released across the striatum. And this will lead to a range of symptoms, including motor and non-motor symptoms. And a lot of it's down to the loss of dopamine in the striatum, and it's progressive over time. We have an effective symptomatic relief for many years from L-DOPA, but we've been giving patients L-DOPA in the clinic for about 50 years, and it doesn't prevent the progression of the disorder. We need to understand why these neurons are dying and how we can stop them. If you peer into the brain, In a control individual, look at this region called the midbrain where the substantia nigra is. You'll see these dark neuromelanin positive dark cells which make dopamine. Neuromelanin is possibly a protective mechanism against dopamine, which is actually quite toxic itself. So this is a control individual, and look at a patient with Parkinson's. This region, these dopaminergic neurons, the neuromelanin positive neurons have largely gone. And if you peer further into the brain with a microscope, you'll identify this aggregated protein pathology. It's called Lewy body pathology. They're aggregated protein inclusions present in both sporadic and genetic forms of Parkinson's, and they're staining, or if you identify proteins within them, they're proteins such as ubiquitin and this protein alpha-synuclein that form the Lewy bodies. My first PhD student said even to the untrained eye, they look malevolent. If you stare down the microscope at them, they stare back up at you in a pathological specimen section, and they've been thought of as that they're actually killing off neurons. Whether they are killing neurons or whether the formation of these aggregates is the last gasp of a neuron to survive as it sequesters otherwise toxic intermediates, we don't know. But this is too late. This is, post-mortem tissue. This is once neurons are lost. How do we understand the very earliest changes in cell and synapse biology? Synapse is the way the neurons connect and talk to each other. How can we intervene earlier to prevent the cells and synapses from dying? That's the vision. And Parkinson's was first described by James Parkinson, who was a GP working in the East End of London. And he published his first essay on Parkinson's in 1817. So obviously we've just had the 200th anniversary of it. And I said in a sort of professorial way, I said to my students, you should go and read his original paper, it's really good. And then I thought, well, if I'm asking them to do it, I should do it. And I read it and it's absolutely brilliant. So you will not find a better description of why you should do cohort studies, the type that Michel does, than he wrote 200 years ago. He wrote, the diseases of long duration, and to connect, therefore, the symptoms which occur in its later stages, with those which mark its commencement, requires a continuance of observation of the same case, or at least a correct history of its symptoms, even for several years. That's why we run the Monument Discovery cohort, right? And he said it 200 years ago. He described many of the symptoms. He talked about Parkinson's, including symptoms, including a disordered state of stomach and bowels, and the sleep becomes much disturbed, which we know is true. He didn't get everything right. He said on current cures, the intelligent will never fail to avail themselves of any opportunity of making trial of the influence of mercury, which I do not want to recommend. But get this, 200 years ago, he wrote on target and drug discovery, there appears to be sufficient reason for hoping that some remedial process may ere long be discovered by which at least the progress of the disease may be stopped. And 200 years later, we're still trying. So it is a great, very early description of a neurological disease. So is the aging brain just going to shrivel as we age? Is decline inevitable? So on the left here, we have a post-mortem brain of somebody who died healthily and somebody here who's died of Alzheimer's. You can see an atrophy and shriveling of the brain. Is this inevitable? Will over time our brains turn from a plump grape to a shriveled raisin? Well, no. Parkinson's and Alzheimer's and related conditions are not simply a result of aging. Aging is the biggest risk, but it's not inevitable. These are disorders and conditions with specific causes. It's not just aging, and therefore we're confident that if we can understand better why the neurons are dying, we will be able to think of ways to slow down that loss and prevent the loss. And a lot of the understanding of where we are with Parkinson's the last few years has come from the revolutionary impact of genetics. So we are living in an age of genetics and medicine. We have every cell in our body contains the DNA, which is the message that makes up the genetic information that makes us who we are. So there's a code of genetic information made up of these 4 bases, A, C, T, and G. It's 3,000 bases per copy per genome, and there's 2.2 meters of DNA packaged up inside every cell in our body. And this is conveying the information through the generations that makes us who we are. The whole genome was first sequenced in 2003, over 20 years ago. We've been understanding it more and more since. So this revolution in medicine started in 1950. 1953, when James Watson and Francis Crick first described the structure of DNA. And this is their very famous wire model that they made in Cambridge. And they're standing there, Francis Crick and James Watson, pointing at the model in 1953. 50 years later, to celebrate the first sequencing of the human genome, they were reassembled around their favorite model, pointing at it. So it's absolutely, you know, the DNA story of discovery was why I became a molecular biologist, because it's a fantastic story, and it's absolutely revolutionized the way that we're able to do our science. So genetic information has absolutely revolutionized the research understanding of medicine and disease. By understanding our genetics, it allows us to understand and predict to some extent who is more likely to suffer from a disease, and this will, we hope, contribute to earlier diagnosis, earlier treatments, which either delay or prevent the disease. So about 10% of Parkinson's is genuinely purely genetic. So mum or dad may have Parkinson's and it's passed through. The other 90% is what's called a sporadic disorder, or idiopathic, which is just a fancy way of saying no known cause. But the other 90% has a genetic susceptibility to component to it. So the genes that contribute to susceptibility, to likelihood to get it, that then combines with aging and with unknown environmental factors, toxins, lifestyle, we really don't understand, to contribute the likelihood to get Parkinson's. So genetics is one part that we use extensively. The other part that of work that we use is stem cells. So we're on the right page. Here's an advanced lecture slide on what is a stem cell. So a lot of what we know about stem cell biology comes from early experiments such as this. This unfortunate individual is Prometheus, who made the terrible mistake of stealing the secret of fire from Zeus and giving it to mortal man. Big mistake. Zeus got very annoyed, and Prometheus was condemned to be chained to a rock, and every evening an eagle would come down and peck out his liver. Thanks to the tremendous regenerative capacity of stem cells, the liver was then able to repopulate and regrow until the next evening. The eagle came down and did it again, and it would be still happening if he wasn't saved as one of these tasks of Hercules. However, data from experiments such as this lead us to the conclusion that stem cells are cells that can divide without limit, stem cells can regenerate tissues, and stem cells can make a whole range of different cell types. So the idea that we could make perhaps stem cells from individuals is very exciting. So for a long time, stem cells have been understood in the context of development. So everything, at least in Western cultures, goes left to right. So you have stem cells, and in development, the stem cells, embryonic stem cells, would divide and produce all the cells that make up the developing fetus. And in all of us, there are adult stem cells, and many of our organs, such as the intestine, skin, that turn over all the time, involved in tissue repair. And this was how it was. Until this chap, Shinya Yamanaka, came along, published a revolutionary paper in 2006, in which he said, why can't you do this? Why can't you take body cells, and the cell type he worked with was skin fibroblasts, introduce some various cellular genetic trickery into them, that we used 4 genes, which he expressed using a virus to carry the genes into cells, that reprogram body cells, in this case fibroblasts, back into stem cells. Now, this came out in 2006. It was a paper with only 2 authors on it, right? And it's a phenomenal amount of work in the paper. There are 2 of them on the papers. They must have worked amazingly hard to get it to work. So they were able to show then this conversion of a skin cell to what we call an induced, because you've made it, pluripotent, because it can make many cell types, stem cell, iPSC, induced pluripotent stem cell. And this work, Shinya Yamanaka and John Gurdon, who'd done a lot of the earlier pioneering work on stem cell biology, got the Nobel Prize. 6 years from discovery to prize is lightning speed for the Nobel Prize Committee. And it has absolutely revolutionized work in understanding human diseases, particularly in a neurological disease where you can't get brain biopsies, right? You can't take evidence out of somebody's brain and study it in the dish. You might be able to do that for liver or muscle, for example, but where you've got difficulty in accessing living human brain tissue in life, it's been revolutionary. So we can take these fibroblast cells, reprogram those into what we call these iPSCs, induced pluripotent stem cell, And then we can turn them into any cell type we like. We could make cardiomyocytes if we're interested in heart, but we're interested in neural cells. We want to make dopamine neurons, and enables us now to do this from people with Parkinson's and from healthy individuals. So why do we want to do this? So the reason we want to grow Parkinson's neurons in the lab through stem cells is we want to understand the cellular pathways that lead to Parkinson's, find what we call the cellular phenotype, find differences in cell biology that are different between dopaminergic neurons from Parkinson's individuals and healthy control cells, and understand why these neurons are different. In humans, it's taken maybe 50 or 60 years for the neurons to die in Parkinson's. My PhD students do not want 50-year projects. So we're not waiting for the neurons to die in the dish. We just want to understand why they're a little bit different and whether that sets up the likelihood that they will die later. Do you remember I said 10% of Parkinson's was genuinely genetic, 90% is of unknown cause but has genetic influence? We can study the rare genetic cases to give us a window into the cell biology of most Parkinson's. And so we know through various studies that many of the cell biology features are in common between the rare genetic forms and the common sporadic forms. So we study the genetic forms. So we study stem cell lines made from patients who have various mutations that cause Parkinson's. The other thing we can do is really exciting, and I'll come to this at actually the very last slide, is that we can work in the lab with cell biology and stem cell samples made from patients, individuals that are walking around as part of Michelle's clinic, because she'll have taken detailed clinical notes, and we'll be able to understand how the detailed clinical notes of the individuals that accompany the cell samples reflect the differences we see in the lab. And it's called disease stratification. How can we understand how different patients have different duration or length or type, and how do we match up the clinical notes with the notes that we have in the lab? And we can then use these cells to what we call screen for therapeutic molecules. And I'll show an example a little later where we understand some of the things that are different between the Parkinson's neurons and the healthy neurons. Can we find drugs or small molecules which could correct this cellular deficit right the way through to drugs? And we have benefited from support from this government platform, the UK Dementia Platform Stem Cell Centre, which we opened a few years ago. So this is how we go about it. So this is Michelle taking a skin biopsy from one of the first patients in the discovery cohort who made a donation of a skin biopsy. And then we can take this skin biopsy back to the lab, chop the skin biopsy into little bits, and it's a lump of skin here, and the fibroblast cells will grow out from the lump of skin. We have a fantastic colleague who works in the Dunn School of Pathology called Martin Cowell, and her team for many years has been doing this reprogramming, turning the fibroblasts into stem cells, and then my lab has then turned these stem cells into dopamine neurons. And this is the technology, the method which allow us to study patient neurons in the dish and compare the biology of these neurons between the Parkinson's patients, typically with genetic mutations in this case, to controls. And so what does it look like? Well, this is a lump of skin that's sitting in the bottom of the dish. And it's over time you can follow how this grows and changes. So you can see that this is the lump of skin here, and the fibroblasts, these elongated cells, start to grow out of the lump of skin and spread across the Petri dish in the lab. And eventually they'll fill up the dish, the little 6 centimeter dish that you're working with. You can then harvest these fibroblasts and then transduce them with these viral gene vectors that contain genes that reprogram the fibroblasts into stem cells. And stem cells grow in these sort of molehill-like clumps. So this is a group of cells to be reprogrammed to form stem cells, and they're growing other fibroblasts around them, maybe dying off, but these have become stem cells and they'll grow. And then what we do is to take the stem cells and then differentiate them, turn them, convince them to become neurons, and you start to add various neuronal growth factors, and these cells start to send out little long projections, which you can see down the microscope. The whole process takes about 60 days, and eventually the stem cells here will have sent out these, become neurons and sent out these electrical wires, if you like, these axons that make neuronal connections across the bottom of your dish. And it's really exciting. You can then stain them for various markers that prove they are dopamine neurons. So these are markers, quite see the light, but that's okay. There's some, we were able to stain these neurons with different proteins in different colors. Beta-3 tubulin, marker that marks all neurons. pH in green is marking some of these neurons as making dopamine neurons. You can see the different cells stained. These neurons will make dopamine, which is good, and they'll make more dopamine when you give them L-DOPA. These neurons are electrically active. That's what neurons do. When you stimulate them, they fire electrical signals. The longer you take them, the more signals they'll send, up to 10 weeks and beyond. And they will also signal with calcium as well. We'll come back to this a little later. As you grow these neurons over time, they have very rhythmic calcium pacemaking activity that's typical of dopamine neurons. And a lot of the work that we were doing a few years ago, this is all funded by Parkinson's UK, and the BBC found out about it and wanted to come and film it and do some work. And it's great because it's charity funded work. It's really good to help raise the profile of the charity Parkinson's UK. And so this is Michelle taking the skin sample, and then this is Palab Ghosh, the BBC Science correspondent, standing in front of the camera, opening and closing an empty Petri dish, which isn't particularly useful. But the guy Sam Everts behind him, the technician, is cutting the sample up into the in the dish to allow the cells to go out. This technique where we stain proteins in the cell is called immunocytochemistry. I think there should be more immunocytochemistry on primetime news. So I was really pleased when these images went out on the 6 and 10 o'clock news, and they came to do interviews as well, because it helps raise the profile of Parkinson's UK and the charity and our work. So that's a kind of introduction to some of the technologies that we use, and then a few examples of some of the work that we do. So one of the aspects of neuronal biology we work on is calcium. Calcium is not just all about teeth and bones. It's an extremely important cellular signaling molecule that helps cells and neurons in particular work the way they should. So calcium is an important signaling molecule within the cell. So different concentrations of calcium exist in different parts of the cell, and that helps activate different cellular processes. Calcium enters the cell across the cell membrane through calcium channels, and the differences in concentration are often very high. So the cell has to use a lot of energy to pump calcium around. It's metabolically very expensive and exhausting pumping calcium around. Dopamine neurons use a lot of energy and are vulnerable to exhaustion because they're pumping this calcium in and out. And then they pump calcium between cellular organelles. So organelles are bits inside the cell that have very specialist function, and they're often pumping calcium around between them in a very important way. So neurons have two ends, and that's super important to realize. So this is actually a mouse brain, not a human brain, but the idea is the same. This is the substantia nigra, where the cell body is here. This is the nucleus. This is where the cell body, the nucleus, the genetic information sits here in substantia nigra. And then they send out these long axons across the brain from the substantia nigra into the striatum. So this is the axons projecting into the striatum here. This is where a lot of the electrical activity of the neuron originates in the cell body. And then almost miles away, if you think about it, down the other end is these axons where the dopamine is then released. So this is the cell. This is the cell body. This is where the what's called tick over occurs. These cells are constantly ticking away. There's calcium is being pumped across the plasma membrane here, and this calcium pumping is accompanied by electrical activity. These neurons firing away down this end, and the electrical signals are transferred all this way along the axons. The dopamine is released when the electrical signal gets to the end, and when the electrical signal gets to the end of the axon, calcium comes in, and this is the signal for the release of dopamine neurons. So the more you study dopaminergic neurons, the more you don't just wonder why they die in Parkinson's. You ask yourself, how on earth do they work at all? So human dopaminergic neurons are enormous arborizations, extensive branching. So if you add up all the bits of a single dopaminergic neuron, it'd be 4 meters long, and makes 2 million synapses. This is over 10 times more connections than any other neuron is asked to make. And yet they have to do this. So they produce electrical signal here, send electrical signals down the end, and then the dopamine is released at this end.
Okay, fine.
So all along the axon, calcium is fluxing in and out of the organelles, the aspects of cell biology, all the way along these axons, all the way along to the end. So these dopaminergic neurons are constantly active. They're constantly producing electrical activity. They're constantly producing dopamine and calcium flux. They're always on. It's a bit like sitting with your engine idling. You're in gear, your foot's on the clutch, so you're not moving anywhere, but your foot is flat on the accelerator. These neurons are teared up, fired, ready to go. Why would you want a cell type that's constantly on edge, ready to go? Rah! That's why. These neurons trigger movement, right? And you need a cell type that's constantly ready to go. The minute you require to move, because a saber-toothed tiger is jumping out at you, you move. So these neurons are constantly on edge, ready to move, get us out of trouble. And it's because they're constantly been on edge for a long time that they're vulnerable. When you get their bioenergetic stress across the body, it's often the dopaminergic neurons which will be the first to die when the mitochondria stop working, because they've been evolving to get us away from saber-toothed tigers. So we're trying to understand how calcium moves around a cell. So these are two organelles within a cell. This is the mitochondria, the part of the cell that makes energy and power. And this is an adjacent organelle called the endoplasmic reticulum, or ER. And this is the cell membrane. This is inside, that's outside. So we have calcium moving in and out of the cell membrane, and calcium moving out of the ER into the mitochondria and fluxing between the two. And understanding how calcium moves and how that's regulated, I think, is important to understand dopaminergic neuron function. And so this is work by Maria Claudia in our lab, who's made these genetically encoded reporters that can allow us to trace calcium around the cell. So this reporter here is in the ER. And when you see the intensity lost, that's because calcium is moving out of the ER. And it moves across and into the mitochondria. So this is actually the same image, and you can overlay them and show that calcium leaving the ER is entering the mitochondria. Once you have the way to record and measure how calcium is moving between organelles inside neurons like this, you can then see how a Parkinson's neuron will handle calcium differently from a healthy neuron. So we study neurons that contain genetic variants in a number of different genes, and one is called GBA. So this is working with dopaminergic neurons that contain a GBA mutation in a Parkinson's patient compared to control neurons. And the red is the GBA and the black is control. And this is measuring calcium release from the ER. And you can see the release of the of calcium from the GBA patient line is lower because the red is lower. And then the calcium moves across into the mitochondria, and you can see the uptake into the mitochondria is less. So once you have an assay like this, in which you know that your Parkinson's neurons are different, in this case in handling calcium, you can then move this onto, this is the phenotype. This is an aspect of cell biology that's different. We can then use that as a phenotypic screen. We've took these patient neurons from a GBA patient, and we screened 4,000 compounds from AstraZeneca against this set of neurons, which we grow in tiny little multi-well plates, and we measure the ability of potential compounds in this AstraZeneca drug library to correct this deficit in calcium signaling back to normal. And we found a few hits, and we're working on what those might be, and the way those compounds might work, and the way the targets might work. And we've just written to Michael J. Fox last week for a bit more money to follow that up. So once you've got neurons in the dish that are different between patients and controls, you can screen against potential drugs that might correct that. So to sort of move towards a wrap up, it's complicated inside a cell. So there are multiple cellular processes that contribute to Parkinson's, and we're trying to understand the different bits of cell biology that are important. So probably the first thing that happens is we have a protein such as alpha-synuclein, which is usually recycled through a process called autophagy, which is run by lysosomes. So usually proteins that need to be recycled or reused, they've got damaged, they're processed by this system called by lysosomes, which are acidic organelles inside the cell that usually recycle the proteins. But as we age, lysosomes become less acidic and they become less effective at chewing up proteins, aggregated proteins, and the aggregated proteins perhaps start to deposit inside the neuron, or if the cells can't process and break these aggregated proteins down, they often throw them out, chuck them out of the neuron. These neurons have to cope with this failure to process aggregated protein correctly. They had spending their time pumping the calcium around, which we've just talked about as an exhausting activity because the differences in concentration between the inside and the outside of the cell. They also have to handle dopamine, which is a terrible, nasty cytotoxic chemical. They release dopamine, and then when it's inside the cell, they package it up into vesicles to protect it from the rest of the cell from the dopamine. Mitochondria start to fail in Parkinson's, and usually when mitochondria start to fail, they get processed by lysosomes, but the lysosomes are becoming less acidic, so they're not able to process the mitochondria, so dysfunctional mitochondria will build up, leading to oxidative stress. And many of the genes that we study, such as GBA, LRRK2, alpha-synuclein, PINK1, and Parkin, these are genes mutated in relatively rare forms of familial Parkinson's. They map onto this pathway that tells us some of the best places to look for potential aspects of cell biology to correct. So, we've studied this a lot now in the lab for maybe nearly 7 or 8 years or so, making stem cell neurons and asking what's different between Parkinson's neurons and healthy control neurons. We've shown the Parkinson's neurons have a reduced electrical activity. They fire less electrical signals on stimulation. They release less dopamine. So we've shown that the neurons from healthy neurons release more dopamine in stimulation than the Parkinson's ones do. The mitochondria don't work so well in Parkinson's neurons. There's a deficit in energy generation, and there's various deficits in cell biology. The aggregated proteins are less effectively cleared through the autophagy process in Parkinson's neurons. So all of these things are different and changed. One thing we'd like to do in the lab is to try to get human neurons doing what people study in mouse neurons. So if you want to study the beautiful, intricate architecture of dopaminergic neurons and how it may change in disease, the mouse models are really good. This is the individual nigal neuron. As I said, it branches, and each neuron will innervate about 6% of the striatum, this amazing arborization adding up to meters long. And we'd like to do the same with our human neurons. And you might not be able to see it very well because of the light, but what we've been able to do is to grow human neurons in these chambers where you grow the cell bodies here, that's the equivalent of here, and then you send the neuronal projections of these human neurons down microgrooves, and then when they reach the other side, the other end of this channel, they produce these exuberant arborizations that we think are sort of mapping similarly to those you see in the mouse. And if you can do this with human neurons, you can start to put different human neurons together to model this basal ganglia circuitry, these different neurons talking to each other as they do in a brain. And we published a paper earlier this year that attempted to do this. So in the human brain here, you have 3 different neuron types in the basic circuit that starts to fail in Parkinson's. You've got cortical neurons here growing from the cortex of the brain on the outside down to the striatum. You have dopaminergic neurons. These ones are projecting into the striatum, and they meet on this striatal neuron type called spiny projection neurons. So it's when these 3 neuron types meet in the brain, it's this interaction here that's critical for Parkinson's. So we've now managed to grow these different cell types from human neurons together in these chambers. So you have cortical neurons here in our dish, which grow through these channels and into this bit here. We've got the dopaminergic neurons that project in a similar way along the dish, growing up here, and then they meet and converge on these neurons called spiny projection neurons, which they have these spines along their length, and these different 3 cell types converge, And we showed that the spiny projection neurons, they grow better, they mature better when they get input from these other neuron types. So what we did is we put these neurons together and worked out how the spiny projection neurons functioned. And then we replaced in this circuit the dopamine neurons from a healthy control individual from a patient with Parkinson's. And when these dopamine neurons here, looking like this, were from a Parkinson's patient, the spiny projection neurons had changes in the biology. So we're trying to understand how these different neuron types can talk to each other and why that's different in Parkinson's. Another really cool thing we're doing is trying to make these circuits of these different neuron types, making these tiny little microgroove channels, but we're making the whole thing out of oil, which is really cool. So we have these two colleagues in engineering who've built this robot here. And what it does is it is able to make these tiny little channels and grooves and chambers out of oil. And we can have neurons that grow on one chamber here, and they send their neuron projections through the gap into the other side as well. So we managed to get neurons that grow in a chamber and project through to the other chamber. And then what we've been doing is, this is neuron outgrowth, and then we can cut the neuron, because you can just sort of cut through with the robot through the oil walls, and then you watch how the neurons repair. So we've sort of put different neuron types together in these dumbbells, with one neuron type projecting into another, and then we've added in a third chamber, so we call a trumbell, and put the dopamine neurons, the cortical neurons, and the striatal neurons together, all built by our robot. And it's again a nice example of being working with engineers to make these multiple chambered devices. Drug discovery is hard going, right? It's a really difficult process. Of course it is, otherwise we'd have all discovered the drugs by now. But it's difficult and it's getting harder. So drug discovery takes a very long time, from your initial ideas of potential target to make the molecules to target that target, moving it through drug discovery, preclinical, and the different clinical phases of 1, 2, and 3, through to FDA or Federal Drug Administration approval, at least in the States. It's expensive, takes 10 years and $1 billion to take this progress through, and it's getting harder. So this is a quite infamous graph in the field with different bits of data on. This is the spending on R&D in pharma companies over 10 years. You can see it going up. This is the number of applications for new drugs, i.e. flat. And this is the number of approved drugs down here, i.e. flat, despite the spending going up. And you can see a lot lower approved drugs than the applications for drugs. So it's getting harder. Now, part of that is because if you do a drug trial where you put all patients together in a trial without understanding the different subtleties in disorders or disease they may have, some of the individuals in that trial may have responded very well and some not responded at all, but the whole trial fails because those who didn't respond, their effect dilutes out, if you like, those that did respond, and you'll lose the drug altogether. So in order to better understand how different, say, Parkinson's is between individuals, you need what you call disease stratification. You need to be able to understand how individuals with a condition like Parkinson's are very different. And this is exactly what the work that Michelle and I have been doing will come to in fruition, if you like. So Michelle's cohort is funded now through to 2026. So it'll be, 16 years of longitudinal study of up to 1,000 patients, whole range of biomarker data. We're looking at CSF and blood and proteomics, and looking at alpha-synuclein. We look at DNA. We have a program in brain imaging. We do electrical recordings of brain activity to monitor for sleep disturbance. We do cognitive measurements. We have smartphones and smartwatches in patients that can capture motor disorder features. So a huge amount of data collected from our cohort. And then, of course, we also have the cell samples. So what Michelle's work has done, you might be familiar with it, is her detailed longitudinal phenotyping and cohort analysis has clustered Parkinson's into at least 4 different subtypes, which she's called cluster 1 to 4. And what we have from many of these individuals is we have patient fibroblasts, so we can grow these skin cells and study them, or we can turn these patient fibroblasts into dopaminergic neurons. And we have the stem cell derived neurons, and we have the fibroblasts from many of the different individuals across these four clusters. And this is a very crude cartoon showing two different organelles, aspects of components of the cell, a lysosome and a mitochondria. And there's some suggestion that some of the Parkinson's, which is more motoric, more motor driven, but perhaps slower progressing, might be driven by mitochondrial dysfunction, because Parkinson's that progresses more rapidly, includes changes in cognition and memory, that might be driven by lysosome dysfunction. And the individuals within these clusters, or the clusters, can differentiate those who have a more slow form, which we think might be mitochondrial, and a more rapid form, which we think might be lysosomal. And the reason this matters, if you want to design a therapy against, say, lysosomes, you wouldn't then go and put in your clinical trial for your lysosome targeted therapy Parkinson's patients who have changes in mitochondria, because you wouldn't get the right treatment. So we want to try to test the idea that some of these clusters are driven by dysfunction in lysosomes, some in mitochondria, stratify in detail the patients, and then in a clinical trial, you'll be able to give the right patients the right drug, which is more hope to be successful. So I'll finish on the slide I started with. This is what we've managed to put together in Oxford, and much of which with your help and with Parkinson's UK support, and we're continuing. There's many different funding sources and streams coming from different places, but the fundamental vision that you can only build a translational program when you get together the clinicians and the scientists with different skills who will talk and trust and like each other and communicate. And this is what we're still doing. So thank you very much for listening. Very happy to answer any questions.
Fantastic talk, thank you. I have a friend who has Parkinson's who lives up in York, and we kind of exchange information from time to time. And he sent me this text this week, and I wonder if you could give your thoughts to what he had to say. He's talking about nettles, and he says it seems that nettles both dissolve Lewy bodies and create new stem cells in the substantia nigra, which differentiate into dopamigenic cells. Nettles also contain silica, which is bioavailable, and once in the blood, binds with aluminium to detoxify.
I don't know anything about nettles, but what is certainly true, and when we did our compound screening against cellular changes, a lot of the things that you find are things that we eat all the time. So things that come up in the screen, sort of extracts of olives and mushrooms, and it's just, what's amazing is how much we are of what we eat, and the world around us, be it mushrooms, olives, or nettles. And a lot of the things out there in the environment may have an impact on protecting us against disease. I haven't heard the one about nettles, but I don't know. I don't know, but I mean, just a lot of plant extracts, things out there that we eat and come in contact with may well influence all sorts of conditions, but I'm afraid I can't educate your friend about nettles. Or you.
Like many people with Parkinson's, I have bouts of extreme fatigue when it's hard to get around. And when I feel better, it's past, I tell myself this fairy story, which is that some of your brain cells have died, and it's taken them 10 days or a fortnight or whatever it is to find another way round through your brain. Now, could there be any truth in this, or is it just a fairy story?
Well, what certainly is true is the brain is very good at compensating through various sort of flexible way that neurons work. So when you start losing the dopaminergic neurons, we don't know, because we don't start measuring early enough. But by the time Parkinson's has progressed such as you present to a neurologist, you probably lost about 70% of them, which means the brain has been able to cope and rewire and compensate for the progressive loss, say through the sort of 30s, 40s, 50%, and then it can't compensate anymore, and then symptoms will develop. But we don't yet know enough about how that compensation happens. If we did, it might be something we could harness. So that's kind of, progression and compensation over longer term. I don't know how it might work over a 10-day period, but in a long-term way that our brains work does compensate against the development of Parkinson's probably for many years, many decades.
Can you hear me?
Yeah.
Richard, a fantastic talk. I'm only grateful that I'm not sitting A-level neurology paper or something in here tomorrow. This exam tomorrow. Serious questions. The pandemic showed us that it was possible to speed up research and development of vaccines. I mean, this is incredibly complicated, but from sitting where I am, I'm sure many people in this room would agree with me, the pace of change seems glacial in terms of patients. So if you had a completely free rein, what would you do to speed this up? Because you could you do it quicker? Not as you, but the whole Parkinson's world. And linked to that, what do you think about the American move to pass bills in Congress to cure Parkinson's? Would that help in the UK?
So COVID was an extraordinary case in which, lots of things that usually, as you say, were quite glacial, were kind of put to one side, that we had to get something to work. We had to get something to work quicker. One thing perhaps that the COVID trials had in favor is COVID developed pretty fast, right? And you were able to show an efficacy for a vaccine or a treatment, within weeks or months, which we know is different for Parkinson's, which progresses very slowly. So one big question in Parkinson's clinical trials is how long do you need to run them for before you see an effect? And it depends what your endpoint is. If your endpoint is a slower deterioration, say in motor phenotypes, and you have to measure that by going to see a clinician in a hospital, that may take a long time. But I think for me, the two revolutions are going to happen or are happening in clinical trials. One is patient stratification. So you put into a trial patients who you think have Parkinson's for the same reason. And that means it's much more likely to get a positive result from your compound, which you've identified and picked because it targets the pathways that you think are going wrong in that patient. And the other is the use of digital devices and smartphones and stuff to collect data. So you can, see as we've just heard an hour or so ago, a neurologist once every 18 months or whatever, and it might be the same in the trial. If you have one of those smartphones, smartwatches, you can enter data, movement data, reaction time data on tremor and movement and posture and gait several times a day, day after day for weeks. And the depth and the richness of that clinical data that's collected on the smartphones with voice recordings and all the rest is extraordinary. And that will speed up clinical trials and make them more sensitive. And the placebo effect goes away. So often if you're on a clinical trial, you may be getting the placebo, but you'll feel better. You'll feel looked after. You'll feel more engaged, and you will get better or improve your symptoms, which masks the effect of the drug. If it's a faceless smartphone rather than a charming doctor who's you're engaging with, the placebo effect largely disappears. So I think the advent of stratified clinical trials in which the right patients go in who will respond to the drug better, and the advent of digital data collection will make the trials process much faster. There is no shortage of compounds or potential ideas entering trials, but I think some of them might have worked or should have worked. We just weren't good enough at running the trials. So, and the final thought is, this speeding up of everything to get a COVID vaccine was the right thing to do. Maybe some of the side effects perhaps were underestimated and are now coming out perhaps in some of the reports on the AZ vector virus, but it was what we needed at the time and it saved millions of lives. But I think COVID was a kind of rather special case, but it does show that, yes, if you're focused and you get people together, you can get things through quickly. And I think the clinical trials hopefully will advance and that compounds will be shown to be efficacious if they are.
Thanks very much. It was a great talk. I'm going to ask a layperson's question. You may have answered it in your talk and I might have missed it. I think it might be towards the end. So you were talking about the, working backwards from the stem cell, which I found really interesting. And I just wondered if those dopamine neurons, not from the control group, but from the Parkinson's group, do you find differences, different biomarkers? Have you already got to that point where, person X, person Y are both donating, to give the stem cells, and then you're turning them into dopamine neurons? Right. Have you got to the point where you have differentiated that they're dopamine neurons? Does that make sense, what I'm saying?
So what we, maybe I didn't understand, what we've definitely done a lot of now is to take the stem cells from the patients and their controls and make them into dopaminergic neurons and look at how they make dopamine and how they function.
Yes, so are those particular dopamine neurons, have you managed to see if there's any differentiation between them?
Yes, between patients and controls.
Yes, between patient and control, but
yeah.
between Parkinson's patients.
Between different Parkinson's
yes.
patients. Okay. Yes, the genetic cases. So, so for instance, is the names of the genes in red are the genes that you often find mutated in Parkinson's. So, for example, if you take neurons from patients who have a mutation in GBA, you'll find a lot of misfolded protein aggregation, ER stress, and lysosome dysfunction, because GBA is a lysosomal protein. If you study Parkinson's patients who have mutations in PINK1 and Parkin, you will see deficits in mitochondria. And LRRK2 is another gene that's involved in lysosome dysfunction. So in some cases, you can find deficits in dopaminergic neurons from different individuals because of the mutation that they carry. We've started to do it in the sporadic patients who carry no particular mutation, but you need more of them because the effects are going to be weaker or less because they don't have a thumping great strong genetic mutation. But a lot of what we know of where these genes fit on this pathway, for example, PINK1 and Parkin being around mitochondria, is because they do have differences, because the patients have a different genetic mutation from each other, so they look different in the neurons.
Right, okay. And then one last question, which is probably totally unrelated, okay, but just out there in terms of, are there any links between Parkinson's and ADHD relating to the dopamine? I don't know if there's any link at all, or that's too big a question, or
it's a bit too big a question.
Right, okay.
But I mean, the dopamine system does so much more than movement. It's involved in reward and addiction and all sorts of things. So don't think there are direct links.
First question, how do you induce Parkinson's in a mouse? And whatever the technique is, does it give a clue as to what induces it in humans?
What a brilliant question. So there are two ways that you can go around modeling Parkinson's in a mouse. The first thing you can say is, okay, a Parkinson's patient has changes in gait and balance and coordination and the like because the dopaminergic neurons are lost. So you can take a neurotoxin, which will kill off just the dopaminergic neurons, give that to a mouse, wait 2 or 3 weeks. That toxin will kill off the dopaminergic neurons and the mouse will get wobbly, and you can measure the way the mouse is wobbly. What that won't tell you at all about is how the neurons were lost, because we don't get Parkinson's by having neurotoxins injected into our brain, and it doesn't lose them in 3 weeks. But what it does is create a mouse that has a deficit in the dopamine system, and then you can try and correct that. The way that we've tried to do it, which we think is much more relevant, is to take many of these genes and put them in the mouse. And that we found, particularly this gene alpha-synuclein, if you, alpha-synuclein can lead to Parkinson's in genetic cases by the alpha-synuclein gene being multiplied. So a few Parkinson's patients in the world, a handful of families, will have a duplication or a triplication of the alpha-synuclein gene. The protein is perfectly normal, it's just there's too much of it. And that too much alpha-synuclein in these rare families will lead to a genetic early onset form of Parkinson's. So we thought, if you take the alpha-synuclein gene, the human one, put it in a mouse, make a genetic engineered mouse that overexpresses this, it will get Parkinson's, and it does. And you can see that you can model the different features of the mouse. You can look at the way that dopamine is processed and released. You can look at electrical activity. You can follow mice in mazes and make them try and balance on narrow rods. And you can see that they lose balance and coordination over time, up to 18 months. And it's a slow and progressive form of early Parkinson's that we think is a very good model. And we deposited at a research center called the Jackson Labs in America, and it's been taken up by many researchers and companies around the world as well, because the idea of being, if you've got a mouse that has a slowly progressing form of Parkinson's and you can find a therapy which will stop that, that might then be applicable to people. So you're left with a situation where we study human neurons, because in the end it's humans who get the condition, but we do need to study the whole brain so we can study mice as well. And that's why we've got both the mouse work and the cell work as key cornerstones of our program.
Thank you for the talk. One of the things that struck me was that there seemed to be a focus in your talk on the development of systems using which you will be able to look at small molecule pharma. What's your view about the possibility for direct implant of reprogrammed stem cells?
That's a really good question. So we strategically took the decision a little while ago when we started, was we couldn't do everything. One thing we were not going to do, because we as labs and scientists didn't have a proven expertise in it, was transplantation of cells. But what you can do is take the mouse that we've described, the first mouse, which has lost its dopaminergic neurons, and reimplant them, and it works tremendously well, and that's now been taken forward into human trials. The early human trials used stem cells and dopaminergic neurons from aborted human fetuses, and it didn't work very well because it was quite difficult to coordinate the availability of donor tissue to the availability of a patient, and the sort of short number of hours you had to harvest this tissue and put it into the patient. What absolutely revolutionized it was the use of the stem cells, the induced pluripotent stem cells, where you can have a bank of very well characterized stem cell derived neurons, which you could implant. And there's a lot of money and a lot of effort into industrialized neurosurgery. You know, we're doing a lot of drilling and a lot of injections and placing these neurons inside the brain. And yeah, time will tell. It's quite a difficult study to do in terms of the controls, because it's generally not easy to justify ethically the perfect control, which is to take a patient, drill a hole in the brain through complex stereotactic surgery, squirt in a bit of salt solution and come out again, and compare that person with somebody who's got the transplantation of stem cells, because that's a lot to ask a control group to go through. So but some of that work is now ongoing. I think you do it because that control group will get the transplants a year or so later after they've taken their role as a control. So a lot of it is ongoing, and I let's hope it's successful. Of course, we don't mind which of these approaches is successful, and I think a lot of the work is going on the transplantation. We're not doing it, but watch this space. There's a lot of people investing a lot of effort that it will be successful.
Thank you. Thank you very much. Greatly appreciate your talk. If you don't mind, could you just repeat the lysosome versus the mitochondrial in terms of treating them? Because you said lysosome was one certain set and mitochondrial, if you don't mind. Just thank you.
So as with all important things in life, it starts with genetics. So if you have a mutation in a gene like GBA, which is a very important lysosomal protein, those patients tend to get a more rapidly progressing form of Parkinson's that includes cognitive changes and memory changes. That's from the rare genetic cases. If you study people with the rare genetic cases of PINK1 and Parkin mutations, they have mitochondrial dysfunction that actually progresses really very slowly. So Parkinson's may start in the 20s or 30s, but can be very well controlled for decades by L-DOPA because it progresses very slowly. That's what we know about the rare genetic cases. So then the argument is, well, maybe people who don't have a genetic mutation, but have a rapidly progressing early cognitive change, memory change, Parkinson's, might have changes in other lysosome proteins. Whereas Parkinson's patients who have a slow progressing, doesn't really develop very much at all after onset, might have changes in mitochondrial function. That's just a hypothesis, we don't know it. If it was true, you would then be able to say, well, these patients have Parkinson's because of a lysosome dysfunction. These patients have Parkinson's from mitochondrial dysfunction. You go back into your lab and find a therapy that benefits lysosomes. You give it to the patient with a lysosome dysfunction, and you're more likely to see a successful clinical outcome in your trial because you've stratified the patients. If you give the same drug to everybody, these patients do really well, these patients don't respond, you pool all the data, and your effect disappears, right? And once the drug's failed once, it's kind of gone. So that's why it's better stratification of the patients. You give the right patients the right drug, and then you monitor daily, regularly with clever digital devices.